Hello, everyone, and welcome to today's Cell and Gene Live, Advancing Gene Delivery, LNPs, Adenovirus, Lentivirus, HSV, and more. We're delighted to have you join us for what promises to be an engaging and insightful discussion. It's my privilege to feature our two distinguished panelists, Dr. Francesca Barone, Chief Scientific Officer at Candel Therapeutics, and Dr. Karen Kozarsky, President of Vector BioPartners. Both of them bring a wealth of experience and expertise in their respective fields, and I'm thrilled that they're here to share their perspectives with us. Throughout our discussion, we encourage you to participate by submitting your questions for our panelists using the Q&A tool, as Heather said. Some of our best questions and coverage points come from our audience, and we will address as many as possible during the session, so please do submit them as you think of them. And so thank you again for joining us, and let's get started. So first up, I'd like to kind of level set our conversation here, as I always do on our Cell and Gene Live events. And so Francesca, I'd like to start with you. Looking at whether it be lentivirus, adeno, HSV, LNP, what have you, what delivery platform do you believe is currently the most underutilized, and perhaps what needs to change, whether it be technically or commercially, to unlock their full potential?
I think that, first of all, I can talk for our experience. We developed viral immunotherapies for solid tumors. So we use a lot both adenovirus and HSV platforms. I think that both of them could be utilized more. There has been a little bit of a stuck in the development of these therapies because of the concept of intratumoral delivery. I think that that a lot of people in big pharmas felt that in terms of development is more difficult to deliver therapies directly to the tumors, and therefore has been sort of slowing down the process of development. Now there are companies, even big companies like J&J, for example, that is demonstrating a renewed interest in the intratumoral delivery. It's actually a very smart way of delivering therapies directly to tumors and without exposing the overall body to an extensive toxicity. So I would say both adeno and HSVs, and in general, oncolytic viruses or viruses that induce a strong response in the immune system are a little bit underutilized and could be utilized more if we overcome this limit of the intratumoral delivery.
Great, thank you. And we will be getting certainly to HSV later on in our discussion, adeno as well. Before we do, Karen, I'd like to get your perspective on the same thing. When you're looking at all of our delivery platforms, what do you believe is currently the most underutilized, and how can we change that?
Yeah, so looking at these particular platforms, it's really use of lentivirus in vivo. So as many of you know, there are multiple lentivirus products that are being developed or on the market as ex vivo gene therapies, where you treat the patient's cells with the lentivirus and then reinfuse them, and which is very effective for certain diseases, but is a complex process. So being able to use them in vivo and have them target the appropriate cell types is currently under development in many places. And that just it's a continually evolving field with trying to target them appropriately in vivo, getting them efficient enough and to the right cell types. So that is just one area that could be really more utilized and would certainly increase the use of lentiviral vectors.
Thank you. And so, there we go. It looks like lipid nanoparticles received about 50% of our vote here, which we will be getting to later in our discussion, but just an interesting point. I want to start with lentiviral vectors. And Karen, this is probably a good discussion for me and you. To kick things off, I'd really like to get your take on where lentiviral vectors stand today in gene therapy, especially as the field works through some important scientific, safety, and manufacturing questions. And so first up, what are the key scientific or clinical rationales today for choosing lentiviral vectors over other delivery modalities, particularly in ex vivo versus in vivo?
Yeah. So as I mentioned just before, it's been primarily used for ex vivo applications. And so what defines it for any disease you want to treat, you have to figure out, of course, which cell types you want to target And that helps you identify what's your best tool to get there. So the way the toolbox is, there's no single vector type delivery vehicle that will work in every cell type, at least not yet. So, so for lentiviral vectors in particular, what it's very strong at is transducing dividing cells. And because it typically integrates into the genome, that then gets passed on to the daughter cells. So cells of the immune system, for example, are a really good target for that. Especially, you can isolate them ex vivo, transduce them with lentivirus and reinfuse them back into patients. So that's an area that can be very useful. In vivo applications, it's a little bit harder. You need to use typically a more localized delivery, but still, as I said, people are starting to develop vectors to target more carefully. It also has a relatively large cargo capacity. So I know it's all perspective. So my perspective, I've spent a lot of my career working with AAV vectors that has, 4.5 kilobase limit of your cargo. And for lentiviral vectors, it's significantly larger than that, more like 8 kb. So that's another area where you might want to shift to a different, to a vector such as lentivirus.